Hi everyone, how are you doing? Hello, can you hear me?
Hi, Dr. Kaur. Yes, we can hear you.
Dr. Kaur. Hi, Vinay, thank you. All right, well, uh, good number of students. Good evening, Jasmine. All right, any specific questions that you may have for me? Sorry, I'm late. I was at school. We had the event today, the research symposium. John was there and probably few more students. Any specific questions?
If no one does, is it possible to go over again regarding the linear acceleration and the angular acceleration for the vestibulocochlear system?
Yes, yes, definitely. All right, let me share the screen. I think it will be easier to explain that way. Um, One second. I got some questions together for you guys for tomorrow morning. We will practice together. They are from the TA reviews, basically. I thought it will be good use of the time. We all can learn together. So I'll bring those questions and we'll solve them together tomorrow morning. Okay. So you can see my screen, Diego?
Yes, I can.
Okay. So basically, you all are clear about the cochlear stuff. We are good on that, Diego?
As in like the audition part, yes. It's more like the vestibular system that I'm
Yeah.
having a little
Okay.
confusion, especially on the linear acceleration.
Yeah. So basically, Um, so this one is fine, like you understand for the ampullae part, the cupula, the kinocilium, stereocilia.
Yeah,
Yes?
so you essentially rotate your head, but the liquid lags behind, so that actually creates the displacement, and whatever the cilia is actually pushed away or towards their kinetocilia. That's where it gets either polarized, depolarized, and that gets transduced, right?
Yes, yes, yes. So
But then that
in
one,
the yeah, in the linear one, I mean, don't confuse yourself a lot here. Basically, all this is telling you is if you are moving your head forward in this one, since these otolith stuff that is sitting up there on this gelatinous mass or mucopolysaccharides that you see down here, and that's where your cilia are they are sitting there, right? So if you are moving your head forward, you basically bending your cilia, right? So if you are bending the cilia and that opens up the channels and this is gonna depolarize. Does it make sense?
yeah. So it's essentially gravity controlled. The thing is that I guess, failing to visualize how is this system at rest, because on the diagram, the utricle and the macular kind of like difficult to identify. So in the diagram on the left, for example, I understand that there will be like the three projections for the three vestibular systems, but then I don't understand which one is the one that is laying flat or the one that should be flat at rest.
So this one is so basically when it's not moving, they are not moving. So if they are moving forward, they are depolarizing, right? So if they are sitting straight, none of the channels are open.
Okay, so it is at rest is well at rest, right?
Yeah, rest is
And they have
rest.
the same
Yeah.
system as with the kinetocilia or like the large cilia thing. that they have like one that is larger than the other, and that's what determines the orientation?
Yeah, so basically, it's more of a movement of stereocilia that you should be focusing on this one. I cannot myself, I could not find a better picture where they are really showing the kinocilium in this one. The other one, for sure, in the cochlea, there is a big cilium that we talked about earlier, where all the stereocilia are moving towards or away. Same thing with the semicircular canals, the ampullae that we discussed, right? Here you have a big
Uh-huh.
kinocilium, even though we call it kinocilium. I mean, if you look at word kinetic,
Yeah, of
that
movement.
should be the mobility, but it's opposite here. So basically, it's
Mm,
immobile. But anyhow, whatever. But here, I couldn't find any picture where they really had a kinocilium. So just assume that it's the movement of the stereocilia along with the otoliths, which are heavier in this scenario. Okay,
got it. Thank you so much.
you're welcome. Anyone else? Any other questions?
Dr. Kaur, I have a question.
Yes.
Can we just go over like what we need to know for the olfactory pathway?
Yeah, sure. Um, So basically, if we talk about olfaction or you talk about the taste receptors, all you need to know is how do the channels open up. You see my point? Remember, I was talking to you golf and camp for olfaction.
Yes.
So let me go there if I can find. Okay. I don't have the other figure here, but anyways, let's talk here. So basically, your odorants, they are talking here to the receptors, right? So something is in the air here, which is binding to these receptors, and then multiple. So receptors are only near about 1,000 in number. But your nose can pick up, as I mentioned earlier, there are near about 1 trillion scents that you can pick up. So that means each receptor can sense multiple odors. Is it making sense?
Yes.
So there are a couple of receptors that these odorants will be binding to, and together they will produce that particular smell. Is it clear?
Yes. Makes sense.
Yeah. And now, how this receptor is transducing, that is the second question. So, first is like how a cross fiber pattern is generated, that is like the odorants, they are binding to the receptors, and the whole smell deal is being sensed. But how the receptors are really transducing this electrical signal is that these odorants, they are basically binding to the G receptors here.
Mm.
And then they're gonna activate the adenylate cyclase CAMP pathway, which is gonna open the cation channels further. And that's how the depolarization of this will happen. And that will be passed on to the fibers further down.
Okay.
Yes, clear?
Yes. That makes sense. Yeah.
Okay, great. So
Thank you.
not just you're welcome, not just for the olfaction, but you have to be ready whether we are talking about bitter or sweet. Remember, we talked about again the G proteins there. Those are the different G proteins which are working there. They're gonna work through IP3 calcium pathway, and they will open those TRP channels that we discussed earlier, the transient receptor. potential channels to get the cations in. If we talk about the salt, how salt is getting picked up by your tongue receptors, that is going to be through the ENaC channels, epithelial sodium channels. And as I mentioned earlier, the protons, since we are just following Costanzo, for that one, I'm not changing at this point because then it becomes, kind of heterogeneity across campuses if other faculty are not transmitting the same information. So we're just going by ENaC channels for now, how Costanzo is writing there. But for your knowledge sake, I had told you there is a recent reporting, and this is through the PubMed data that I had found few papers where they are saying that Otopetrin 1, those are the channels which let hydrogen ions pass through in order to let you have the taste of a sour thing or basically acidic material. Does it make sense?
Yes.
Okay. All right. Any other question?
Other than knowing the simple or like the triggering mechanism for the different flavors, and their channel, we don't really need to go over again the IP3 channel, the IP3/calcium pathway, right?
For which one, Diego?
For like all of the tastes. So I don't know. So let's say sweet. We just need to know that it is a G coupled protein, and then that
Yeah,
it does the
yeah,
IP3 calcium
yeah. Yeah, you don't have
cascade.
to go through more than that. Yeah. I mean, we have studied that so many times now.
Yeah.
So, I mean, by this time we assume that you know it. And just up to this much, if you know how the whole cascade is working and how the transduction is taking place.
Yeah, I was more concerned like once you reach like one of the PK molecules and then like a transcription factor and all of
Yeah,
that. If we needed to know
yeah,
that.
no, yeah, you don't have to go into that detail.
Got
My
it.
only point was to tell you, see, that if the transduction has to happen, that means if any taste is present there or any smell is present there, how your receptors are picking that information and changing into the electrical signal. And by now, you all know that if your neurons have to carry any current or change any current on the neuronal fibers, that means cell has to be depolarized or some channels have to be opening up. And those channels are gated by some kind of energy, right? Whether it comes through opening of a sodium channel, which is camp gated, whether it is, be helped by IP3 calcium pathway, whether it is being helped by some CGMP like we did in case of the eye vision transduction, right? Remember, CGMP was guarding the sodium channels there. So that was my point of telling you, like how that neurophysiology would occur and the transduction of that signal would take place. Make sense, Diego?
Yeah, got it.
Everyone else, are you guys clear or any questions regarding this?
Yeah, I wanted to ask a question about that. So for because I asked the TAs and they told me to follow up with you. For the sour one, the
Mhm.
hydrogen blocks the voltage gated correct channels for potassium, and then that's what causes the depolarization.
So, yeah, I mean, there are several theories, Miss. Yeah, that's what I was telling you guys. In DO class, we had little bit more discussion than what we had in MS class the other day. So one of the DO students had asked this specifically. He emailed me,
Mhm.
and he was wondering, like, why don't we have the clear pathway outlined here? There are several ways certain papers they report, like what you are mentioning,
Yes, ma'am.
potassium and hydrogen
Mhm.
interaction, and that's how the protons are getting in. Um, and or the autopatrin, what I was mentioning earlier, lately, it's a recent paper that came out in 2023, Cell and Science. I think it's one of the science journals, very hi-fi journal. So good paper. And that suggests that these autopatrin receptors, they basically let hydrogen ions in, and that's how we perceive the sour taste.
Okay.
But there is no clear guidance till now. I mean, it's getting more and more clear. For the autopatrin one, seemed more of a pathway that one could follow. But
Mhm.
unfortunately, I was telling the DO class as well, and I'm telling you all, we all gonna follow what Costanzo has written, which may not be right at this point.
Mhm.
Hydrogen ions they are showing going through ENaC channels, which cannot be true. Those are specifically for sodium, as the name suggests. Those are epithelial sodium channels. They let sodium in. Um, but for this exam's sake, we're gonna keep the information same because of
Okay.
the discrepancy. If I tell you something else and other campuses are not getting the same information, then I'm in trouble for it.
Oh, I agree. That's not fair to them, so I understand.
Yeah. So, but basically, they say that very recent discovery. You can look into it on the PubMed.
Mhm.
There is a paper out there. It says autopetrin 1. Those are the receptors which let hydrogen ions in, and that's
Mhm.
how you perceive it.
Okay.
But just between you and I mean, you guys and I was just bringing the information in, but sometimes other faculty would say, okay, if we didn't give the same information, don't give students this and that. And, I
Mhm.
just didn't want to get into all those controversies. So, yeah, we're just gonna follow Costanzo at this point.
Yes, ma'am.
Yeah, it's wrong, but we can't do anything about it. Okay. Anything else? How's the prep going? I feel things I told you earlier, how much technical we were in cardio. Okay, if venous blood is coming this way, what's gonna be your cardiac output, or difficult equations of palm or renal, different segments of renal, what is happening with diuretics and all that. Like those things were very technical, or endocrine reproduction, all that stuff, GI. Those were like some technical stuff there. Here, I think it's straightforward, but still, if you guys have questions, feel free to ask me.
Hello, ma'am.
Yes, Parambir?
Yes, I had a quick request. Could you go over some of the examples for calculating sound pressure? I just wanted to make sure that I had that I have that down.
Sound pressure?
Yeah, the a decibel equals 20 times log P over P0.
We don't have that in our. You don't need to do any of that stuff for us.
Oh, it wasn't the reading. That's why I asked. Okay, so never mind.
Yeah, I mean, just for the sake of like how much your ear can handle, and that's about it. So it's very loud, then, above 70, decibels, it starts to become kind of, it can bring some damage. Like 100 decibels or 120 is already painful and it will bring the damage and the pain together. Yeah, that much only. We're not going more than that.
Got it. Thank you, ma'am.
You're welcome.
For the physio component, sorry, back to the taste pathway, do we need to stay? I know they've already tested us on the cranial nerves and the ganglion, that
Mhm.
nucleus. Is that coming back for the physio portion, or is that just
No,
staying?
no,
Okay, perfect.
no. I'm just gonna stick with physiology.
Okay, because I know there's one slide you have from class that has a pathway on it. Um,
Which one are you saying, Lucia?
there's one slide, I'll read it, hold on real quick, that you have a pathway scademic, or let me see, where is it on? It's number, it's for the ears though, it's not for the TICE. It's
Oh,
auditory
okay.
transmission.
Auditory pathway?
Yes, the for the ears. Yeah, this one.
This one? No,
Yes.
you don't have to.
Oh,
This is not in our learning objectives. And it's very anatomical, if you ask me.
that's what I was just like, let me just check
Uh,
because I wasn't
you
sure.
may have questions from neuro
Yes.
from this, but not from me.
Okay.
If it was, then I would have already gone into the detail in class.
Okay.
I thought that I covered everything pretty much, but I'm just gonna say it again since we don't have supplemental stuff for this portion. So be ready with all the clicker questions. What I went with you guys. Finish all the TA review questions, the fresh one that he prepared, and we picked up all the older questions from previous tier reviews, and that's what I'm going to bring tomorrow. So we will practice together tomorrow morning when we do our supplemental or mini review or whatever. So I
Okay.
put those questions together for you guys. I picked them up from the previous ones.
So would you like for us to for physiology, I'm just making this clear, to because there was some cranial nerve questions in the one Mr.
I know, I saw that.
Okay, that's why I asked about the tracks and stuff because I
Yeah,
was
I saw that. No, I'm not asking cranial nerve questions. No.
okay. Thank you.
Yeah. Yeah, I removed actually when I prepared the file today for tomorrow. I removed those questions. So I will tell clearly tomorrow again one more time. So when more students are there. But anyways, you can tell your friends I'm not asking cranial nerves or anything. Just clear physiology portion. Okay. Skylar has a question. Skylar?
Oh, yes. I just wanted to ask a clarifying question. This was, I think, something Diego asked earlier, but for example, if someone's rotating their head to the right and then it depolarizes on the right side, but then when it stops, does it flip and then it hyperpolarizes on the right and then it depolarizes on the left?
So it's just going straight, basically. So if you are moving your head to right, one thing that let me go to the figure and I will explain you. Somebody had come to me today the in my office and I described them. Give me 1 second. Let me go to the figure and let me explain. Where did it go? Hmm. Yeah, all the questions that we have in Clicker, please make sure that you finish them all. So here, let's say that if you moved your head to left, let's say, and I just said that left side, you made a movement. So stereocilia, they are moving towards the kinocilium on the left side. And right here is cupula, right? Cupula is all a gelatinous mass that is sitting there. So cupula is basically bringing this movement for the stereocilia to move towards the kinocilium at this point, and it depolarized the whole thing, right? We are talking about the left side now. So stereocilia moved towards the kinocilium and they opened up the pores and things go inside into the hair cells, right? But by this time, don't forget, this was your ampulla, right, where the cupula was sitting. Don't forget that there is endolymph. Now, by the time we move to the left, now endolymph will be catching up because cupula moved faster, right? And it brought this movement and everything, pores opened and channels opened and sodium or whatever cations they're getting in, right? Now, by this time, endolymph is catching up. It's gonna bring that cupula back in, or the stereocilia back into the straight position, how they should be. Is it making sense, Kyla?
That makes so much sense. Thank you so much, Dr. Kaur.
You're welcome. You're welcome. Any other question? Yes, Sadaf?
Dr. Kaur, do you suggest that we study the book as well, or if you think if we just go over the clickers and the PowerPoints that would
No,
be
book for sure. Book for sure. Never ignore the book.
Okay.
Yeah.
Thank you.
Book for sure. It's just the practicing questions or anything that I added extra, and both the tier reviews have to be finished nicely.
Perfect. Also, the slides, some of them do not have any like spark notes under them. Are you going to like add them, or it's just the way that you have posted it?
Some of the slides did have, let me see, some of the speaker notes I had added later, and I think I posted these slides before my clicker session. So I can add the new one.
Okay, thank you so much.
Yeah. But there are some. So there are some slides which have the speaker notes.
Yeah, some of them do, especially the ones that we're talking about the muscles. But like some of the not clicker, I mean the poll everywhere questions, they did not have like explanation.
So polls everywhere, that takes away the speaker notes.
Okay.
You're gonna find them wherever my other slides are, regular slides are. The polls everywhere, that removes the speaker notes in case
Okay.
there were any. So that's why you must have seen I left like this. I have left this kind of slide in there even after the polls everywhere.
Okay.
I have left those, so you may find down there. But I think one or two slides that I may added some speaker notes lately, like the autopatron stuff and some other stuff. I may replace the slides tomorrow morning. Yes, Alina, you have.
Thank you, Dr. Kaur.
You're welcome, Sadaf. Alina?
Hi, Dr. Kaur. Thank you for staying up this late to help us. I just had a question that I want to jump off of that Skylar asked. I'm a little confused. So I just want some clarification. So if the head is turning left and the stereocilia goes towards the kinocilium, so that's depolarized, but then later it goes back to straight, which means it's moving away from the kinocilium. Does that mean it's being hyperpolarized in that moment back to normal to go back to rest, or
it's going back to rest. It's not moving into the other direction. If it's moving to the other direction, it's just going back to the straight, its normal position. It's not hyperpolarizing.
Okay.
Hyperpolarizing means that when it's moving away from kinocilium. That would be your other side of the ear, where
Okay.
the hyperpolarization has taken place.
And this is all happening during the motion of turning your head left and keeping it left?
Yeah,
Okay. Okay,
yeah.
sounds good.
Mhm.
Thank you.
You're welcome. Yes, Diego?
Once the rotation of the head ends and the lymph actually catches up with the rotation,
Mhm.
do we need to, like, that's it, that just returns everything to rest,
Yeah,
right?
yeah. That is good enough for you guys to know. I
Got
mean,
it. Okay.
don't confuse yourself more than that because I think you guys are thinking too much on it. So just remember if stereocilia moved, it already transduced the information, right?
Oh, and sorry, one more question. When we said that in the lymph is more like ICF than ECF, yet we are actually exposing the hair cells to something that it is also ICF. There is still the influx of ions for the depolarization. So should we assume that even though it's more closer to the ion balance on the ICF, there is still a higher concentration outside than inside of the cell? So that we have
For
like an
which
actual
one?
potential
For which one? For which ion are you
for
talking?
for all of them, right? I mean, like we said that the endolymph is more like ICF than it is ECF, right?
That is just for the cochlear stuff.
Oh, okay, got it.
It's just for the cochlear stuff. That's only the endolymph which is there, that is potassium rich. I'm
Mhm.
gonna bring a figure tomorrow for you guys to have little bit more information tomorrow in supplemental, where we'll talk about perilymph and endolymph more. Okay,
Great, thank you.
but for now, just think, see, uh, the movement of the cilia. You are thinking about it. But one point that you guys are not connecting to that, like that movement towards the kinocilium opened some channels and the ions are already in. So the depolarization that had to take place to tell the brain that this movement has happened, it's done. The transduction has already been done, right? Now, if it moves to the right place where it should be, it's going to close the channels, right? So your transduction had already happened, and now we're going to move back, go to the straight position, and that's it, right? Clear, Diego?
Yeah, I'll think about it more as a mechanical receptor, mechanical gate.
Yes. Yeah. Anything else? Yeah, today was a long day for us. We had a research symposium, by the way. It was really good because my students got the first prize for the experimental research. So I'm very proud of them and very happy. Yes, thank you. Um, all right. Doesn't seem there is any other question. So we can meet tomorrow at 11 and come prepared so you can answer questions. We just gonna do questions, practice questions tomorrow. Okay?
Dr. Kaur, I think there's a question in the chat.
Oh, okay. Let me see. What is it? Skylar is asking, where would we be able to find these slides with the questions and answers? Or the one that I'm doing tomorrow, Skylar?
Oh, I just meant the one that you have presenting right now, Dr. Kaur.
Oh, these ones are already up on Canvas, Skylar.
Oh, really? Okay, perfect. Thank you.
Yeah, these are already up there. I thought I put it already. Did anyone check? Sadaf, have you checked?
Yes, Dr. Kaur, they're already there. Today then when I was studying it, there was like about 46 slides.
Yeah.
Oh, I see it now. Thank you so much.
Okay, perfect. I thought I did post it last week, but then I didn't check after. So, yeah, tomorrow's the questions that I'm bringing tomorrow, those are already Jackson sent those to you guys, the TA reviews. One was fresh review that I'm not doing with you guys, but I'm picking questions from the older TA reviews and putting them together. So I'll bring those for practice tomorrow. Okay? All right. Have a good night, everyone. And
Thank you, Dr. Kaur.
thank you. And let's meet at 11 tomorrow.
Thank you.
Bye for now. Thank you.